In this review, we're going to go over, very briefly, some functions of proteins. We're going to go into detail about enzymes, and we're going to review mutations. So, I'm going to talk about 5 functions of proteins. The first function is going to be hormones. Some proteins function as hormones. They're created in one part of the body, sent out of those cells into the bloodstream, where they go to another part of the body, and they send some message to those cells in the other part of the body. So they act as chemical messengers. Some proteins help make up some structure. For example, collagen helps attach muscle to bone, and structurally, it is made up of proteins. Some proteins are integrated throughout the cell membrane, and they help in transportation. They allow some materials into the cell or out of the cell, and we'll talk more about the cell membrane and the proteins in the cell membrane coming up. Some proteins are enzymes. I'll get more into detail with this in the next few slides. And finally, as a last resort, protein can be broken down into amino acids for energy. The body does not prefer to use protein as its main energy source. It uses carbohydrates, but desperate times will call for desperate measures. The function of enzymes is to speed up chemical reactions, and enzymes can be reused over and over and over again, makes them a catalyst, speeding up chemical reactions. It's important to note that without enzymes, the chemical reactions necessary for an organism to live would be far too slow. So the chemical reactions would still occur, but not as quickly as the organism needs them to. So life as we know it wouldn't exist without enzymes. The structure of enzymes, so it's a protein, so it'll have a very specific shape. It has an active site, which is the location on the enzyme, on the protein, where the reaction takes place. And then we have a substrate, which is what the enzyme is working on. And each substrate has a specific shape that corresponds with the shape of the enzyme. So the enzyme can only act on this substrate, on one substrate. So here you can see the enzyme has a specific shape. It acts on 2 substrates, and it combines them together through a chemical reaction to create a product. Here we have 2 different enzymes, each with different shapes, so they act on 2 very different substrates. A round substrate in this example cannot bond to a substrate with a square active site. So enzymes are very specific to substrates. Enzymes function best in certain conditions. Those conditions are temperature and pH. So depending on the enzyme, it's different for each enzyme. The pH or the temperature that they function best at will be different. So for example, the enzymes functioning in your stomach function better at a lower pH. If the pH of the stomach goes too basic, if the pH increases too much, then the enzymes will not function as well. And as you can see in the pictures, the further you get away from the optimal pH, the less the enzyme functions. So same for temperature. The further that the environment is away from the optimal condition, the less the enzyme functions. And the reason is the enzyme becomes denatured. It loses its structure, so it can no longer bind to the substrate because it just doesn't fit. The shape of the protein has changed and it no longer matches up with the substrate, so those chemical reactions don't happen. So before I get into a review of mutations, I want to review exactly what a gene is. So all of your DNA is coiled up, wrapped around itself, wrapped around proteins into a chromosome. So you have hundreds of genes on one chromosome. So if something were to happen to part of the chromosome, you could potentially lose the genes for hundreds of traits, for hundreds of proteins. So one part of the DNA is a gene. Okay, a section of the DNA that codes for a trait is a gene. So that DNA is transcribed into mRNA, which is then translated into a protein. And remember, proteins do everything in your body. If you don't have a specific protein because something has happened to that gene, it could potentially harm you or harm the organism. So there are two general types of mutations. Mutations that happen to the chromosome, so they happen on a larger scale, or a mutation that happens to one specific gene at one specific point on the DNA. Chromosomal mutation. So each of these just signify a section of the chromosome. So section A of the chromosome, section B. So here you can see that section B has been deleted. So we call this a deletion, plain and simple. And the effects of that are negative. You're missing potentially hundreds more genes that code for a whole bunch of proteins. Missing a whole bunch of proteins is a huge problem. Especially depending on what those genes coded for, what specific proteins they coded for, and what those proteins did in the body. So a deletion is a big problem, very negative. In this example, we have a duplication. So section B of the chromosome has been duplicated. This isn't necessarily bad. In fact, they believe that this could actually be a way that some evolutionary mutations have occurred. If there's a mutation that's bad on the second section, not a big deal because you still have the original directions, the original instructions for making the protein in the first section. And then if there's a mutation that's good in that second section, well, then the organism displays that mutation and changes over time. In the third example, we have an inversion. So basically what has happened is part of the chromosome has flipped over. So if you look at the original A, B, C, D, E, F, and then you look at the mutated chromosome, you see that B, C, D, E are backwards. Inversion mutations aren't a big deal. The cell can still read the chromosome, can still read the DNA. And finally, there is a translocation mutation where a piece of a chromosome comes off and attaches itself to a different chromosome. And again, this does not cause a lot of harm to the organism and may never even be noticed until the organism tries to reproduce. So now focusing more in on a specific gene mutation. These are called point mutations. They happen at a specific point on the DNA. So if this is an example of original DNA sequence, there can be substitution mutations. So where this was originally a C, it's now an A. And it doesn't have to be a C to an A. It could be any nucleotide, any base pair changing to any other base pair. It's just plain old substitution. One base pair for another base pair. Then we've got deletion, where a base is completely left out. A base pair is completely left out. And then we have an addition or an insertion, where a base pair is added where it doesn't belong according to the original. So these mutations, these point mutations, have an effect on the protein that's produced. So I wish this picture was a little clearer. It is a great picture, and that's why I'm using it. But I do wish it was a little clearer for you. So if this is the original DNA strand, after transcription, translation, this would be the chain of amino acids, which becomes the protein. During a substitution mutation, there can be a few results of changing a base. So here the example is this C, this first C here has changed to a T. And based on the codon reading, the amino acid was originally a glycine. But now, with the substitution of the T, it's now a serine, and that's going to change the amino acid sequence, which is going to slightly change the protein, potentially. So the effect could be bad. The effect could actually be good, depending on what this new structure does. Something else that could happen is a base pair could be substituted and end up coding during translation for a stop. So that will tell the ribosome or the tRNA to stop coding, to stop reading the mRNA, and it'll actually make the amino acid chain shorter than it's supposed to be. That's called a nonsense mutation. Another thing that can happen is where there was supposed to be a stop codon, it now changes to something that codes for an amino acid, and the protein ends up becoming longer than it's supposed to be. That is a run-on mutation. Finally, and most interestingly, is a silent mutation. If a base pair changes, it's possible, because of the fact that there are 20 amino acids but 64 codons, and there are multiple codons for one amino acid, it could create the exact same amino acid in the amino acid chain, in the polypeptide chain, and then end up creating the exact same protein. So a mutation could happen, and the organism would show absolutely no effects because the protein ended up being exactly the same. And it doesn't really matter in the end. Deletion and insertion can have very drastic effects, especially depending on where in the DNA it happened. So if it happens kind of at the end of the chain, it's really only going to affect the very last amino acid. But the earlier on in the gene, in the DNA sequence that it happens, the more of an effect it'll have on the rest of the chain. And the reason being you can see here in this example, it's, read CAT, 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 CAT. The important thing being that the codons are 3 base pairs long. So if a deletion occurs where this A is removed, well, now it's still read as 3 nucleotides. It's just shifted over to the left. So it's CTC, ATC, 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 and you can see it makes a completely different amino acid. And that'll go on until the very end of the gene. So it can completely change and have a devastating effect on the protein. And the exact same thing for insertion. Okay, so again, the original was read CAT, 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 and made these amino acids. But if you insert, now the reading frame shifts to the right. It's still read every 3 codons. So where it was CAT, it should have been here CAT, but now it's going to be ACA, which creates a different amino acid, TCA, 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 which creates a different amino acid. And that's going to affect the entire rest of the chain. So all of the codons from the point of mutation on are read incorrectly, and it has a massive effect on the protein that's made.